all the teachers and students. Today is National Technology Day. Uh, Department of Computer Science and Mathematics are organizing this webinar. Earlier, we have observed National Science Day on 28th February. This date, this National Technology Day, holds a historical significance as it marks the anniversary of India's successful nuclear test that was carried out in Pokhran, Rajasthan in 1998 with the code name Operation Shakti. If we look into the definition of technology, then you can see that it is the application of scientific knowledge for practical purpose, practical applications. Technology is nothing but it uses the scientific principles and applies them to change the environment or to change the way of human life. We can discuss some remarkable technological innovations like internet. We are talking in the webinar, and you have joined in this webinar through the internet connection. So communication and information exchange globally using the internet. We can talk about smartphones. You are watching this webinar in the smartphone, I guess. So this is the pocket size computers for communication and accessing various informations. We can talk about the invention of electricity. Nowadays, you have observed in our college, principal sir installed sensor-based lights. If you enter into the room, the light will be on and otherwise off. So it saves electricity. So it has enabled the development of modern infrastructure and industries and changing the way of our life. We can talk about the blockchain technology. So it was originally developed for cryptocurrencies like Bitcoin. Later, it has been used in various industries, including finance, supply chain management systems, and healthcare also. Innovations like 3D printing, renewable energy. Also, we can talk about the medical imaging and the AI technology. So innovations of artificial intelligence, everybody is using nowadays ChatGPT, and for medical imaging, all the scientific innovations are being made. Actually, I am working on the medical imaging field where I see I'm actually working on COVID-19 and pneumonia. For X-ray plates, it is in the naked eyes, it is not visible that it is not perceivable that it is a pneumonia or it is COVID-19. So using AI technology, we have achieved a great accuracy with no time. So we uses technologies for information exchange. We can clean our clothes. We can prepare meals. We can go from one place to another. We are using technology in our day-to-day life, like door locks, like different furnitures. But we seems that these are for granted. We seem that this is less impressive than self-driving cars and artificial intelligence. So innovations in machine learning in the field of biomedical research is happening today. And using this artificial intelligence, the particular disease can be diagnosed using the machine learning techniques. So to discuss on this matter, today we have our guest and my friend, Dr. Mosaddegh Hossain. He is assistant professor of Alia University. But before that, I would like to request our principal sir to say a few words on this day.
Can you hear me?
Yes, sir. Yes, sir. You're audible, sir.
So, good morning to all the members On behalf of the cordially welcome Dr. Hussain for taking your time out in this Saturday morning. And it's sometimes for us, and for time and our
I think there is a network issue.
by and our college. Now I can proudly say we are organizing every moment by thanks and thanks to Dr. Ghosh also for having Dr. Ghosh that wonderful topic. Now that is say so but I have got for my students already know that our computer science honors program is reviewed from India. So everybody there is share this for the village that but our computer science and it's a nice way that you're organizing computer science because you cannot imagine a day now, second now, without because that is the raising of just five. And we discussed that the concept of an understanding and block together. All these are coming in big ways and so doing also we are using the spectrum. And medical field is not beyond that. And I personally I was in engineering college with months earlier. And there is biomedical engineering department, and I have seen the application of these ideas of AI and ML, AI and artificial intelligence starting in biomedical analysis of the biomedical data, and I appreciate the importance of something else, diagonalize using the numerical industry for so I hope that in this session, our students come to know what the latest innovation is. So the scheme of pictures and technology startup ignite the innovative mind. That is what we have been doing at our institute also in the past few months. We have organized science foundation, science exhibition, where from here for 5 5 IPS and five more are waiting, and we are also organizing promoting our students in startups. We have already registered one MSME Sumadha last week, and two more are in the pipeline. We are organizing the events. Main objective is to there is very profound youth at our college, and majority of them are girls. So that we can promote them, but not just towards the regular way of the life they have been living. Rather, promote the light of innovation. Then some of them become entrepreneurs. Then they can start up in a properly. Bank loan, and that can only happen if science. And so I request all my science department and the as well as all the art department. So diversity. Let us let the amount of this. And if I wish,
Thank you very much, sir, for your encouraging words. Now I will call upon Dr. Uh, Mossadegh Hossain. I would like to introduce Mossadegh Hossain. Dr. Mossadegh Hossain is currently working as an assistant professor and former head of the Department of Computer Science and Engineering, Alia University, Kolkata. He has earned his doctoral degree from Department of Computer Science and Engineering, University of Kalyani. Dr. Hussain has published several articles in highly reputed international journals like Nature Scientific Reports, Genes, BMC, PLOS ONE, and in many conference proceedings and book chapters. He is also recipient of the prestigious TARE research grant from Department of Science and Technology, Government of India. His research domain includes computational biology, bioinformatics, machine learning, and deep learning. So I do like to welcome Dr. Mossaddeq Hossain.
Am I audible?
Yes, completely fine.
Okay. Thank you, Midul and Principal Sir, for inviting me in this particular program. I'm very I'm very much excited to present this particular talk on analyzing disease progression using gene expression data. Okay. Without further delay, let us try to present my screen to discuss some overview of how actually gene expression data can be utilized for performing disease progression. Okay. So let me just present my screen. So is my screen is visible to you?
Yes, Hossain, carry on. Okay.
Okay. So actually, we will just present an overview of machine learning techniques, how machine learning techniques can be utilized for disease progression. Different types of disease can be, say, for example, Alzheimer's disease, or say, for example, AIDS, or it can be any types of cancers. Okay, using a type of data, biological data known as gene expression. And we actually are computer scientists who develop machine learning and deep learning algorithms for analyze such biological data such that we can say predict, or we can say infer some uh, uh, some problems or some difficulties in gene expression data, such that it becomes easier for the medical personnels to uh, analyze those results to say generates or to some say inspect the results obtained by us through thorough biological experiments. Okay, so before discussing about how the machine learning techniques can be employed for performing such analysis, let me just give an overview of the biological overview of about the gene expression data. So everybody from our biological knowledge know the concept about a gene. Okay, we know that gene actually a small regions inside deoxyribonucleic acids, and it is the molecular unit of heredity. Actually, this gene defines the characteristics of individuals. Okay, say for example, it defines whether a person will be tall or short, whether a person will have say eye color, say black or brown, like that. And everybody every often us actually understood that these biological systems are extremely complex. Okay, at the molecular, at the very basic levels, we have different types of biomolecules, like say gene, protein, RNA, and those actually parts of a larger constituents, and that constituent name is known as cell. And different types of cells different cells constitute a larger component in our body, which is known as a tissue. And the interaction or the coordination different among the different tissues forms different organs of an organisms. Okay. Although I have told you that this gene is the smallest region inside our gene is the smallest biomolecular unit in our body, but the genes uh, um, uh, but genes are different to different cells. Okay, not all the genes are different to different cells. Say in a particular cell, type of cell, say for example we have skin cell, okay, or say for example eye cell or lung cells. In every types of cells we have the same set of genes, okay, and that's set of all genes which are parts of or which constitute a particular say cells is known as a genome. Okay, so genome is the entire set of say biomolecular particles or biomolecules present in a cell, and this set of biomolecules are same in every cells. Okay, so all the set of genes present in here will be present in say liver cell or lung cells, but um, actually not all the genes will be able to produce same amount of uh, proteins or say same amount of another particular biological component which is known as messenger RNA or mRNA, and that leads to different behavior of the cells, and um, that basically change how hair cell acts differently from the lung cells or liver cells like that. And this gene expression is nothing but the amount of messenger RNA produced by the different genes at a particular instance. And there are different, say, biological technologies like, say, DNA microarray or RNA sequencing that are used by biomedical personnel, which can be utilized to simultaneously, say, monitor the expression levels or the amount of messenger RNA produced by different genes at a particular instance. Okay. So let me just tell you what actually gene expression data looks like. Okay, so as I have told you that a particular gene expression is the amount of messenger RNA produced by a particular genes at a particular instance. Okay, so if we actually collect the gene expression data for an entire cell, it will look like a matrix. Okay, so here we can say represents the row headers or the rows using the gene names. Okay, I have just used some identifier names here, and these columns actually indicates the sample conditions or the experimental condition or a particular instance, and therefore a particular element in this matrix, okay, say for example this particular value, say this 43 or say this particular value, say 41 point, say for example this 41.87, this particular value actually indicates the expression profile of this particular gene under this particular instance. Similarly, this particular say value 2.48 indicates the expression profile of this particular gene at this particular instance, or say similarly this particular value indicates the gene expression profile of this particular gene at this particular instance. Okay, so this way we can use the gene expression data to analyze or perform different types of machine learning works, or we can perform different types of machine learning research to say analyze the progression of disease. How it can be done. Before proceeding into that, let me just tell you one say fundamental concept in molecular biology. Okay, how actually the different activities of our body performs. Okay, so according to a concept known as central dogma of molecular biology, which was actually proposed by a scientist Crick during 1950s, that the DNAs produces or the genes produces messenger RNA or mRNA according to a technique known as transcription. Okay, so genes are transcribed into messenger RNA, and that messenger RNA again uh, uh, produces proteins which are larger components in larger biomolecules in our cells through a technique which is known as translation. And the proteins are those biomolecules which are the main constituents or main say components that perform different types of activities in our body. And whenever the amount of messenger RNA produced by a particular gene changes abruptly in our body, okay, say for example a particular gene, okay, say for we have different naming convention for genes, okay, say for example p53 one particular gene, okay, whenever the uh, expression levels or the amount of messenger RNA produced by the p53 gene changes abruptly, then we might have different types of disease, okay, depending upon the amount of changes. So um, uh, uh, as I have told you that differential gene expression is the concept which indicates that not all the genes are equally able to produce the amount of genes at a particular instance. And this particular concept is known as differential gene expression. Okay, so not all the genes are equally expressed or equally able to produce the same amount of genes or same amount of proteins at particular instance, and it is known as differential gene expressions. And using different statistical, mathematical, or say computational techniques, it is possible to measure the amount of changes that occurs between the expression levels of a pair of genes at a particular at a particular pair of experimental conditions. Okay, say for example, whenever say we have a normal cells, okay, normal persons having a specific type of cell, say for example skin cells, then then the expression levels of all the genes present in the skin cells might be some values, might have some values, but whenever say that uh, specific type of say cells, say for example skin cells, will be affected with some disease, say for example skin cancer, then the amount of the expression levels of the genes present in the skin cells will have some different values. Okay. So say we can identify those genes whose expression levels changes abruptly um, in such a pair of experimental conditions, say for example disease affected samples and as well as normal patient samples. Okay. So we can use a kind of plot which is known as volcano plot. Okay, this particular plot is volcano plot to actually visualize the which genes are having abrupt changes or significant changes in their expression profiles at a pair of experimental conditions. Okay, so here say for example here the genes which are say represented using this yellow color and red color, they have their expression levels have been highly say changes or highly regulated in two experimental conditions. Okay, the genes which are indicated with red colors are actually known as upregulated differentially expressed genes, meaning that their expression levels have been highly increased from the base condition, say for example normal condition, into disease states. And here the genes which are indicated using yellow color refers to the genes whose expression levels have been significantly decreased in the say experimental conditions, say for example say cancer stage or any disease state. Okay, so am I audible clearly, and is my screen is visible to you clearly? Okay, so similarly, the genes for which there is no significant changes in gene expression data are indicated using black color. Okay. And although these the genes which are represented using these blue colors are significant D genes, but their expression level not been say, significantly decreased or increased. Okay, so they are represented using blue color. So this type of plot is known as a volcano plot, and we can use it to visualize how actually gene expression changes in different experimental conditions. And um, before discussing about a concept which is known as gene co-expression network or gene expression networks, through which it is possible to perform some machine learning say operations or machine learning tasks to say perform or analyze the progression of different diseases. Let me just give an overview or say recapitulate the concept about a network. Okay. Everybody, you may be familiar with the concept of a network. Actually, a network is interconnection between the nodes. Okay, say for example, whenever we have say internet networks means two computers are connected over the say internet technologies for data transfer. Similarly, say whenever we have say any kind of biological networks, say there are different types of biological networks. Say for example, protein-protein interactions networks or gene co-expression networks, okay, or gene regulatory networks. These are different types of biological networks. So here, say instead of the nodes representing computers, the nodes actually represent different types of biomolecules. Say for example, in a protein-protein interaction networks, the nodes represents the proteins, and the interactions among those proteins, whether a particular proteins say perform some kind of say interactions with another protein, indicates whether there will be any connection in between the proteins or not. Okay, so what I have told you regarding this network. So network can also be represented using say mathematical tool known as a graph. Okay. So you all know that graph can be represented using say this kind of say using this kind of say diagrams, and here say this A, B, C, D, E, these are the nodes, and say an edge or say a link between this A and D indicates that there is a path or interaction among these nodes, say A and D, and we have interactions between this node B and D like that, but we do not have any interactions like say D and C or E and C like that. So similarly, we can say represent this graph using another mathematical concept which is known as adjacency matrix. So here in the adjacency matrix, one will represent that we have a connection between the nodes. Okay, say for example, we have a connection between the nodes, say A and B. So here we have 1. Okay, similarly, we have a connection between A and C, so we have 1 here. But as there is no connection or A's between A and E, so this we have 0 in this particular element. Okay, so using and this is basically one type of say a network which is known as undirected networks, or and as well as say unweighted networks. So whenever we have any interactions or is between two nodes, then we will have 1, otherwise say we will have 0. But we can have different other types of networks. Say for example, we can have directed networks where we have say direction between the edge connections. Okay, so say D interacts with A. Okay, so the edge is from D to A, not from A to D. Okay. And also sometimes we may have weighted networks where instead of say 1 and 0, we'll have the connection strength or say connection, the relationship between the nodes connection. Say for example, say how say in case of biological networks, we might have some absolute value. Say for example, 0. Say 75 or say zero point 0.3, like that. So we'll have say different weights associated with the different ages. So that is known as say weighted networks. Okay, so let me just introduce the concept of gene expression networks, so gene co-expression networks. After that, we have already learned what do we mean by gene expression, and then what is how the gene expression data actually represented, and now we are in a position to say introduce the concept of gene expression networks or gene co-expression networks, which can be utilized or which have been utilized predominantly for say analyzing the different disease progression in computational biology and bioinformatics. So I have already shown you this kind of matrix, say gene expression matrix or gene expression data, which can be represented using this matrix. Okay, so this is a toy example of how gene expression networks or gene co-expression networks can be constructed from raw gene expression data. Okay, so these gene expression data can be available publicly from different biological sources or biological databases, and also it is possible to generate this kind of biological data, gene expression data, using biological experiments. Okay, so this is just a toy example how gene co-expression networks can be constructed. So what we can do, we can just use a particular technique known as correlation. Okay, say for example, we can use the concept of Pearson correlation coefficients to say find out how the say expression profiles, that means the gene expression values of a particular genes under all the sample condition are correlated with another gene. Say for example, how the gene expression profiles or the how the gene expression across across the different samples of gene G1 is related to G7 can be measured, okay, using a technique known as correlation coefficients. And we can use any type of correlation coefficients like say Pearson correlation coefficients or Spearman correlation coefficients or mutual information. There is different say statistical measure through which it is possible. So as say we all know that the correlation among a particular say element to itself is 1. So you can see that this correlation value with correlation coefficient among the gene expression profiles of gene 1 to itself is equal to 1. Okay, and similarly, if you perform say Pearson correlation coefficients between say gene 1 and gene 2, we will have say for example value is equal to say 1 as well. Okay, but the say gene expression values between gene 1 and gene is equal to here say 0.5, meaning that there is a significant difference in the expression profiles across all the samples of gene 1 and G3. See, you can see that the gene expression value for expression for the gene say G1 at sample condition say S1 is equal to 199, whereas in case of G3 it is 957. Similarly, we have a drastic change in gene expression values say across sample S2 for G1 and G2. Okay, so after say finding out this particular say correlation matrix or correlation correlation matrix from the gene expression data, what we can do, we can just say construct a graph using this particular co-expression values. And here node indicates the genes and the age weight indicates their correlation values. Okay, and after that we can select a significant threshold value to say keep the nodes having significant gene expression values to be connected among themselves, and say connected among themselves or retain their connections, and discard the nodes having very less correlation strength. Okay, so is this thing is clear to you, everybody? And am I audible? Is it clear to you? Oh, I have to make full screen, right? Okay. So is now my screen is clearly visible? Okay. So now now what we can do, there are different types of you might be familiar that there are different types of machine learning algorithms. Okay, see, for example, one type of machine learning algorithm is known as unsupervised learning, and the example of such unsupervised learning techniques are clustering and and association rule mining. And there is another type of machine learning algorithm which is known as classification. Okay, so another say supervised learning examples, classifications or say regression analysis. Okay, so both say classification, clusterings, both type of machine learning algorithms can be applied on these kinds of say biological data, biomedical data, to analyze those data to infer some biological critical activities in our body. So in this particular talk, I will mainly focus on a particular machine learning technique which is known as clustering. So you might be familiar with this particular concept that this clustering is nothing but to find out groups, okay, or say clusters or modules based on the similarity and the dissimilarity among the different objects. Okay, so similarity means say for example, whenever suppose you have collected the data, say weight and height data of all the students in a particular class, and you wish to say uh, cluster the students, okay, based on their BMI. Okay, so whether the students having say normal weight or say overweight or say below weight. Okay, so according to that you wish to cluster the students. So in that case, say in that case we can utilize a technique known as clusterings. So what we will do in this particular case, we will try to find out the say correlations among the weight and height values of the different students, and we'll try to group those students having highly similar weight and height ratio. Okay, similarly we can say keep another set of students in another group having similar height and weight. Okay, so this using this particular clustering technique is nothing but to say form or find out groups of say objects based on their similarity and dissimilarity based on different say, features. Okay, say for example here features means say height and weight, okay, of the students. So here we will say we can have different types of such clustering algorithms available in our machine learning. Okay, say for example K-means clustering algorithm, and there is another type of say clustering algorithm which try to find out say a set of nested clusters. Okay, and that particular clustering technique is hierarchical clustering. And in this particular say talk to say keep my say space sort. I am just discussing about a particular technique, hierarchical clustering. Okay, so this hierarchical clustering technique is try to say obtain the clusters or the groups among the objects based on say nested tree-like structures or hierarchy of structures. And there are different say again categories of hierarchical clustering technique. One is known as agglomerative and another one is divisive. Okay, both actually try to find out the clusters or the say groups among the objects, but their way of say, finding out the clusters differs. Okay, say agglomerative clustering techniques actually try to this kind of say whenever we try to find out the clusters using hierarchical clustering technique, that finding out the clustering clusters can be represented using a type of diagram known as dendrogram. Okay, so using this dendrogram we can easily visualize how actually clusters are formed among the different objects. So from this particular diagram we can see that the similarity between object 1 and object 3 are highly similar. Okay, because their say a height length or the distance length is low. Okay, so we here in case of agglomerative hierarchical clustering, what we'll do, we will actually try to say initially consider the individual objects as clusters and then try to merge two similar clusters. So in agglomerative hierarchical clusterings, we will first of all say try to merge two more similar objects. So here 1 and 3 are more similar objects and we merge them together to form the first such nested clusters. Then we find that the similarity between these two objects 2 and 5 are highly similar. Okay, so then what we do, we actually form a clusters by merging these two objects. And then we find that the similarity between these obtained clusters and the object 4 is highly similar. So again, we merge these three objects to form another nested clusters. And finally, later on, these two clusters, 1 and 3, and then the clusters with objects 2, 5, and 4 are merged together to form another bigger clusters. Okay, this particular clusters. And finally, the last object is merged with this particular nested clusters, and this way the agglomerative clustering techniques ends. Okay, so is this thing is clear to you? The concept of hierarchical clustering, agglomerative hierarchical clustering. Anyone having any doubt? Am I audible?
Yes, you are audible. Please carry on.
Okay. So this is the algorithm. I'm not just to keep my talk short. Let me just skipping this. Okay, there are say different types of say algorithms. One such algorithm is known as ACNES. Okay, which was proposed by Kaufman and Rosso. Okay, so there are actually different again categories of different again types of say agglomerative hierarchical clustering algorithms. These actually try to the way they differs actually by the concept that how actually we compute the intercluster distance. Okay, how the distance between two clusters are measured to merge, perform the merge in the next steps. Okay, that differs from one particular technique to another technique. Okay, one is known as single linkage, another known as complete linkage, and another say average linkage. See, in case of single linkage, the distance between two clusters is measured as the minimum distance between a pair of points where one point belongs to one cluster and another point belongs to another cluster. Similarly, here in case of complete linkage, it is the maximum distance, and in case of average linkage, it is the average distance between each pair of objects, where one object belongs to one cluster and another object belongs to another cluster. Okay, and this is just this divisive clustering technique works just the opposite way how I have explained the concept of agglomerative hierarchical clustering. So it's actually initially considered that all objects belong to the same clusters, and then try to actually split the clusters based on based on their dissimilarity. Okay, just the way of thinking or way of working or the working procedure is just the reverse of agglomerative hierarchical clusterings. Okay, so here say I have just to explain this concept, I have just added this say toy diagram. Okay, how actually this Pearson correlation coefficients can be computed. Okay, and represented using say heat map plot. This type of plot is known as heat map. Okay, and then how and I have performed that hierarchical clustering techniques. Okay, agglomerative hierarchical clustering techniques to obtain this set of clusters, and then finally we have obtained different sets of say gene expression or gene co-expression clusters from that. Okay, so here we can see that this these two genes, gene 3 and gene 4, are highly similar, and they forms a single clusters. Okay, what does it implies from this particular say gene from this particular clustering results? This indicates that the expression levels or the expression profiles of this gene 3 and gene 4 are highly similar, and from our biological knowledge we know that actually genes do not perform their activities alone. Okay, they generally try to form or try to perform their activities by say interacting with other genes. Okay, so say for example a particular say biomolecular works, okay, biological works are not being done by a particular by a single genes. Okay, not a single gene is responsible for a particular disease. Okay, so many genes actually interact among themselves to form disease. So this particular so from this kind of say machine learning algorithm techniques, through the say gene expression data, we can actually discover which genes are responsible or which genes act together in certain experimental condition. That means, say for example, what are the genes responsible for acting together for performing different types of biological activities in say for normal persons, and what are the different types of genes which are responsible for performing different types of biological activities whenever that particular person is affected with some disease. Okay, say for example Alzheimer's disease or any type of cancer like that. Okay, so this is the objective of say such kind of say biological or say bioinformatics analysis using machine learning. Okay, so just to keep my talk short, okay, just I will show you a sample sample works, okay, which I have done to analyze the progression characteristics of say AIDS from normal persons to AIDS using the gene expression data. Okay, so everybody we are familiar that there's a person say affected with whenever a person's affected with a particular type of virus known as HIV, then this AIDS actually occurs. Okay, and in case of whenever a person actually say interacted or person say contaminated with say HIV, then it actually affects our immune systems. Okay, so there are different stages in which that HIV progresses. The progression of HIV affected persons stage. And this is a symptomatic stage where the persons may encounter different types of say symptoms in his or her body. Say for example, might have the persons may have symptoms like say fever, or sore throat or rash or say joint pain or muscle pain. Okay, and at this particular stage, actually, one of our say important type of cells known as CD4 plus cells, CD4 plus T cells destroys, which actually affects our immune systems. That's why we have this kind of symptoms in our body. And this acute stage is very short stage, say last around say 3 to 6 weeks, and after that, the HIV affected persons move into another, say, long stage, which is known as chronic stage. And this is generally asymptotic in nature and may last around, say, 8 to 10 years. Although, say, at this particular stage, CD4 plus, the amount of CD4 plus T cells reduces, but that rate of say decreasing the CD4 T plus cells is very low, and also say the amount of HIV viruses replicates at a very low rate at this particular stage. And there is another particular stage which is known as non-progressor stage, which rarely happens whenever a person affected with say HIV virus. Only a small say portions of HIV affected persons may clinically stable or say remain active even without having any kind of treatment or antiretroviral therapy, and say even lives for say 30 to 40 years or even more than that. And normally, say after the chronic stage, most of the persons move into the devastating stage, which is known as say acquired immunodeficiency syndrome or AIDS stage, where the amount of HIV viruses rapidly increases and the amount of CD4 plus T cells decreases significantly, and that's why the our say HIV the body HIV affected person's body fail to protect him or say her from different types of pathogens, and ultimately persons affected with AIDS dies. And here just I have I have say shown us sample results, okay, just a part of our results to say you through a plot to indicates how actually that machine learning techniques can be utilized to analyze the progression of the different stages of that HIV or AIDS disease. So here in this particular plot, I have shown that what I have done that we have obtained the differentially expressed genes across all pair of stages. So, say for example, normal stage to acute stage, normal stage to chronic stage, like that. Yes. 8 slide. Okay, what? Okay, let me see. Then try to. Okay, let me just. Oh, oh, sorry. So let me just try to again stop presenting and then again try to present my screen. So is my screen is visible now? Okay, okay, okay. Actually, very sorry. So actually, these are the slides that I have say gone through. These are the slides. This slide was visible, right? Okay. So let me just control L. Is now it's visible, right? Slide number 9, right? Oh. When I am actually say changing to full screen mode, the slide is not visible. Okay. Yeah, that's the problem. So whether you are facing any kind of problem when I say keep this keep my slides in this position without full screen. Okay. So this is the slide number 9 where I explain the concept of cluster analysis. Okay, introductory concept, and this I have. Why it is creating problem? Okay, let me just try to
Hussein, please off the full screen mode.
full
If
screen
you,
করলাম আমি তাও
আচ্ছা,
হচ্ছে না কেন একটা মিনিট দাঁড়াও আমি আরেকবার জাস্ট করছি
ডিসকানেক্ট করে দিন।
হ্যাঁ নেটওয়ার্কের কিছু ইস্যু হচ্ছে বোধহয় বা দাঁড়াও একটা minute। আমি মানে আসলে আগে বললে আমি প্রথমে ওটা চেক করতাম তখন হ্যাঁ is my screen is now visible? Slide number 9.
Yes. Now go on.
Okay, so this is the introductory concept about the cluster analysis that I have explained. Okay, using this slide only. Okay, but it is not shown unfortunately. Okay, so this is the slide corresponding to that introductory concept about hierarchical clustering, agglomerative, and divisive. And then this particular diagram, through using this particular diagram, actually I have explain the concept about how actually hierarchical clustering works using agglomerative clustering algorithm. Okay, this particular plot is known as dendrogram, and then actually this is the algorithm for agglomerative hierarchical clustering algorithm. So is my screen is now visible, all the slides? Slide number 12 now.
Perfect. Carry on.
So this is the different types of proximity measures to find out the different different types of say clustering algorithm, agglomerative clustering algorithm, single linkage, complete linkage, and average linkage that I have explained. And this is say toy algorithm for divisive clustering algorithm. And then this particular plot I have actually explained how that gene expression data can be utilized to find out the clusters among the genes, okay, to detect or to discover which are the genes which acted together in certain experimental conditions, like say disease states, or in say for normal patients, like that. So skin flowing, right? I'm now in slide number 16.
Yes.
Okay, so these are the different stages of say HIV that I have say explained. Okay, so now this particular plot I was explaining that in this particular say work I have performed different many analysis, but this is just a single plot to give you an overview how this machine learning algorithm, mainly clusterings, can be utilized to say perform disease progression analysis. So what I have done in this particular work is actually I have identified the differentially expressed genes across every pair of disease states. Okay, say for example, say from normal stage to say acute stage, normal to chronic, normal to AIDS, or say acute to AIDS, okay, acute to chronic, like that. Okay. And after identifying those genes, I have just concentrated upon picking up picking up the say expression profiles of those differentially expressed genes across pair of say experimental conditions. Say for example, say I have picked up the differentially expressed genes in control or normal stage, and picked up the expression profiles of differentially expressed genes in another stage, say for example acute stage. Okay, then I have performed that machine learning algorithm clustering to discover the modules in a particular gene expression modules, or say which genes are acting together in a particular stage, say for example control stage or normal stage, or in acute stage, or in say chronic stage, like that. And then we have tried to discover the modules whose expression profiles, the modules in which the expression profiles of genes are highly changed using different statistical technique. Okay, due to limitation of time, I'm not explaining those different types of statistical measure. Okay, there are different such techniques available through which it is possible to detect say whether a particular say module or cluster, okay, or group say present in a particular experimental condition, okay, and not preserved, okay, or not retained in another experimental conditions. So I have detected those modules um, uh, inside which the expression profiles of genes say, get changes from one experimental condition to another experimental conditions. And through this plot, I have actually shown the protein-protein interaction networks. Protein-protein interaction networks are nothing but, say again, another type of biological networks where nodes are the proteins, and if a pair of proteins interact among themselves, then we will have connection between those proteins, and otherwise we will not have any interactions. So here we have tried to obtain or try to say, construct the protein-protein interaction networks, and then identified the immunoregulatory genes, okay, which actually responsible for performing immuno-related immuno-related activities in our body. As I have already told, actually in case of HIV, our immune system is drastically affected. So here in this work, I have discovered those say gene modules in which there are say lots of expression changes in the expression profiles of the immunoregulatory genes, and we have detected several such genes which are actually preserved and not preserved across the different say modules or clusters of say gene expression networks or protein-protein interactions networks. Okay, say for example, what are the genes which say keeps their or retain their expression profiles across the pair of stages like control acute, control chronic, control stage, and drastically changes their expression profiles across a pair of stages. Okay, so this way say we can perform different types of say different other types of analysis to discover the progression characteristics of different diseases, as well as the stages of different diseases, okay, including cancers or any other types of disease. Okay, so after that, it is possible to again say infer some biological characteristics. Okay, if my screen is visible, 18 number slide. Okay, so after that we can infer some say biological knowledge from our say machine learning analysis. Say for example, we can discover what are the say different biological activities performed by the genes present in a particular say modules or particular cluster or say particular group of genes. Okay, and those actually those biological significant analysis, there are various biological significant analysis. Two such biological analysis are known as a gene ontology based analysis and pathway based analysis to say actually infer the properties and functions of the different say gene products or say a set of say genes or a set of genes or set of proteins. And also there is a technique, there is a biological term known as pathway, which actually indicates the final gene products, or what actually a set of genes will finally perform, what final activity will be performed by a particular say set of genes. Okay, so those kind of analysis we can, biological analysis we can say infer from our say machine learning based disease progression analysis, disease progression analysis, and pass to the say biology say biomedical personnel for further say validations. Okay, so with this actually I come to an end of this particular short this overview of say analyzing gene analyzing disease progression through gene expression data. So so if I'm say I'm happy to answer any questions. If you have any doubt, you can ask me.
Thank you very much, Hussein. Your presentation is very good, and we have enriched using your presentation. So, do you have any questions, the audience? Please ask any question.
Yes. Yeah. Um, actually, here this weighted networks. Okay, is my screen is visible? Slide number 7. Slide number 7 is okay. Okay. So actually, as I have told that there are we can categorize these say biological networks or any kind of networks into two categories. Okay, one is say weighted and another one is unweighted. In case of unweighted networks, it is represented using a binary adjacency matrix. Okay, where say one represent we have a connection between a pair of nodes, and 0 represent we do not have any connection between the nodes. Okay, now here in case of gene expression say analysis, what I can do, say if we find that there is a significant similarity in gene expression say data across all the samples between a pair of genes, then we can say assign the age weight, okay, between the interaction among those genes by 1. Otherwise we can put 0. Okay, if we do not have any significant similarity among the gene expression profiles of that pair of genes. Okay, and in case of unweighted networks, okay, so say for example, sorry, this is for the unweighted networks. But in case of weighted networks, what I can do, instead of say having 0, 1, which indicates whether there will be any connection among a pair of nodes or genes, or there will be no connection among the pair of nodes, we will assign some age weight. Okay, so here for example, the age weight between this gene 1 and gene 9 is 1. Okay, that indicates that there is very high similarity across the say gene expression profiles of gene 1 and gene 9. Okay, across all the samples. Say for example, we can see that here whenever the expression profiles, okay, this is actually again say unsigned network. I have considered the unsigned network. There is another concept, signed network and unsigned networks. Okay, so I have considered that whether I have say given same emphasis whenever. Okay, that's why we have 1 in this case as well, say for gene 1 and gene 9. So here we can see that say when the gene expression value for across sample S1 for gene decreases, then it is true for sample 2 as well as in case of sample 3. Okay, so we have 1 here, but in this case, say for example G2 and G3, okay, whenever say gene we do not have such kind of similarity. Okay, so this we although the similarity, we have a similarity, that similarity value is equal to Pearson correlation value is equal to 0.5, but as we have selected a high threshold, say for example most probably I have selected the threshold value is equal to 0.8 here. Okay, so we have say ignored the similarity value below that 0.8. Okay, so whenever we do not have a say similarity value, we have similarity value less than 0.8, we have this we have not considered that significant say similarity in gene expression values, and we have not created any interactions or edges between that nodes. Okay, so this gene expression, these say weights actually indicates the similarity or dissimilarity among the gene expression values across the different samples among a pair of genes. So if this thing is clear to you? Yeah, 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 yeah. Okay, yeah, actually, we in this particular sample work, this is just a toy example, just to introduce the concept how machine learning can be applied. Okay, you are right that it is possible to represent the say matrix or graph or network using different such kind of matrices. But generally, if we work using this gene expression data in order to obtain the gene one kind of say type of say gene express one kind of say modules in our say say bioinformatics say works, that type of modules is known as gene co-expression modules. In this particular type of work, I have tried to find out the gene co-expression modules. So that's why I have say utilized this kind of adjacency matrix, where actually the say these weights indicates the gene these weights actually indicates the correlation coefficients among the gene expression profiles of the gene expression profiles of the different genes. Okay, that's why just I have used here in this case. Okay,
Hussein,
yeah,
I have a small question that you are
mm,
dealing with large number and pool of genes.
yeah.
So how you are managing these genes, and do you using any dimensional reduction techniques
Yeah,
or?
yeah. Yeah,
Hmm.
definitely. Actually, I have say skipped those pre-processing technique. Although say I have say introduced one particular say pre-processing technique known as differential gene expressions. Okay, this is one kind of technique that you can use to reduce the dimensionality of say gene expression data. So everybody might have been familiar with that, that a human body consists of around say 56,000 genes. Okay, now it is not possible to say due to our computational limitation, it is not possible to perform this kind of analysis using that whole set of say 56,000 genes. So uh, you can say perform dimensionality reductions using different types of feature selection algorithm in say statistical or machine learning. Okay, say for example you can use MRMR feature selection algorithms, okay, filter based any feature selection algorithm, JMIM, like that, or you can perform any say PCA or ICA based say dimensionality reduction techniques. Or
Hmm.
there is another kind of technique which is known as differential gene expression, where we try to actually discard those genes for which we do not observe say substantial changes in gene expression patterns or gene expression values across a pair of experimental conditions. Okay, so we will only retain the expression profiles of those genes for which there is significant changes in the expression profiles profiles of the genes. Okay, so either say we will keep only say significant upregulated and downregulated genes gene expressions. Okay, so this way we can reduce the dimension.
Okay, thank you. Audience, if you have any question, then ask to share. Otherwise, we may conclude our session. I would like to ask Dr. Dhruva Chandra Dhali, Director of Sampur Siddheshwari Mahavidyalaya, to conclude the session by vote of thanks. Dhruva, your audio is not clear. So
Yeah. Thank you, Dr. Mridul Ghosh. Good morning to all of you, and congratulations in this auspicious occasion. Today's day of National Technology Day. So it is a great occasion, and today's without technology we cannot survive anymore. So technology was there, and nowadays technology is mostly using, and always everywhere technology is very much needed, and technology always will be there. So without technology we cannot survive, it is definite. In this great occasion, I am honored to express my vote of thanks to our principal sir, Dr. Sabyasachi Sen. Without his permission, it is not possible, or contribution, it is not possible to conduct such a webinar. And also, today's speaker, Dr. Sheikh Muhammad Mosaddeq Hossain, Assistant Professor, Department of Computer Science and Engineering, Alia University, for giving such a wonderful lecture, explaining very nicely all these things relating to gene expression and how technology can be used to analyze genetic expression. And he also very nicely explained cluster analysis, and thereby making phylogenetic analysis, and finally make a nice dendrogram. So we are very much happy, and also I think our audience, our participant will be very useful or very happy with this lecture. Thank you, sir. We are expecting further you for a wonderful lecture. And also, we are very much happy to express our vote of thanks, Department of Computer Science, all these faculty, and also mathematics, and our very important members of IIC and also IQAC, all the participants, of course, without whose participation it is not possible to conduct such a wonderful webinar. Thank you, all of you.
thank you, sir. So we can sign off. So bye. Have a good day.